Hello friends, today we are going to study about amino acids part 2 under composition, structure and function of proteins. So amino acids can act as acids and bases. If we dissolve amino acid in water, we'll get the structure of Zwitter. What is Zwitter? Zwitter means it is the structure in which the amino acid will carry no net charge. That means carboxylic acid in the form of COO minus and amino group in the form of NH3 plus. So one positive and one negative will cancel out each other and this structure carries no net charge. Now this structure can act as an acid or base. What do we mean by acid or base? The substance that can donate proton are acids. The substance that can accept proton are bases. Now from NH3 plus, one H can be donated and this structure then can act as an acid. So after donating H plus, NH3 plus now became NH2 and COO minus. The same structure can also accept a proton and act as a base. So COO minus can accept a proton and get converted into COOH. So a Zwitter ion or amino acid in the form of Zwitter ion when dissolved in water can act either as an acid or as a base. The substances that have such dual nature are amphoteric and known as ampholites. Now what happens if an amino acid consists of one amino group and one carboxyl group? For example, glycine. So what happens at very low pH? Low pH means more of H plus. When H plus is more, or we can say when the pH is very low, that time the amino acid present in the form of fully protonated. Fully protonated means this COO is having H and this NH2 is also having H and so it became NH3 plus. So here also one H is there, here also one H is there. So this form is known as fully protonated form. So at low pH where H plus is more, the predominated form of the amino acid is a fully protonated form. Now as the pH increases means H plus is going down. So when the pH is increasing or H plus is going down, in that situation the COOH will lose a proton and gets converted into COO minus. So at some point of time when COOH donated its proton but NH3 plus has not donated proton, in that condition the amino acid is in the form of Zwitter ion having negative as well as the positive charge. When the pH is increasing more, that means now it is high pH. High pH means low H plus. Low H plus means now this amino acid will again donate a proton from NH3 plus and get converted into NH2. So what we learned is when the pH is low, the predominated form is a fully protonated form, or from the net charge we can see here which form is dominated, plus 1 or the positive. At high pH, which form of amino acid is dominated? The negative form, or we can say the amino acid will have a negative charge at high pH. At low pH, amino acid will have a positive charge. At high pH, amino acid will have a negative charge. Now, the amino acids have a characteristic titration curve. What do we mean by acid-base titration? Acid-base titration means addition or removal of proton. Let's understand this with an example of glycine. Now, when the pH is very low, what is the structure of glycine? One amino end, one carboxyl end, when H, when R. In case of glycine, this R is H. So we can write the structure of glycine as CH2. This is a carboxyl end. This is the amino end. At a very low pH, we just now studied, at a very low pH, the predominated form is a fully protonated form. So this form is a fully protonated form. That means carrying net charge of plus 1. Now as the pH increases at some midpoint, some molecules of glycine will start losing H from the carboxyl terminal. Now glycine will have both the molecules, fully protonated form also and Zwitter ion form also. Why? Because the pH is increasing. So one by one the molecules of glycine will start losing proton from the carboxyl terminal. So at this particular midpoint, or 50% of molecules have donated the protons and 50% of the molecules are about to donate protons. Correct? This midpoint is this pKa in the graph, which is 2.34 for the glycine. As pH will increase more, a point will come which is known as Pi. Now what happens? All the carboxyl terminal donated proton. So the predominant form is the Zwitter ion form. That means no net charge. This point for glycine, which is called Pi or isoelectric point, is 5.97. Now as the pH increases further, now this NH3 plus will start donating proton and a midpoint will come when half of NH3 plus donated proton and half of NH3 plus is about to donate proton. So those who have donated proton will be in this form and those who are about to donate proton is still in Zwitter ion form. And this pH is this pK2, which is 9.60 for glycine. So at very low pH, what is happening? It is a fully protonated form. That means H is present in amino terminal also, H is present with carboxyl terminal also. As the pH increases, that means in the solution H plus is going down. In that situation, the COO or the carboxyl terminal will start losing proton. At some midpoint, half of carboxyl terminal donated proton and half of carboxyl terminal is in a protonated form. And that pH, that midpoint is pKa1, 2.34 for glycine. As pH is increasing more, now all the carboxyl terminal donated proton and all the amino acid is in the form of Zwitter ion, which is Pi 5.97. With further increase in pH, now the amino terminal will also start losing proton. So the pKa2 9.60, when half of amino terminal donated proton and half is still in a Zwitter ion form or about to donate proton. As we increase the pH further, a time will come when all the amino terminal will donate proton. So now for glycine 2 pK, pK1 and pK2, that means it can buffer around 2.34 and 9.60 pH. The buffering range for this glycine will be 1.34 to 3.34 and 8.60 to 10.60. This we already studied in the previous videos where we studied about pH and pKa. This glycine has two buffering zone around 2.34 and around 9.60 pH. Now the titration curve helps us to predict the electric charge of amino acid. Now there is a relationship between the net electric charge on amino acid and the pH of the solution. Now this is the graph of the glycine which we just studied. In this graph, this 5.97, this is Pi. That means isoelectric point, no net charge. For glycine or any amino acid that contain only one amino terminal and one carboxyl terminal, we can calculate the Pi using the formula Pi is equals to half of pK1 plus pK2, 5.97, which is shown here in the graph. Now, if pH is more than Pi, that means this area, the predominant form will be the negative form. If pH is less than Pi, lower part, that means the positive charge will be the predominated charge. Now, amino acids differ in their acid-base property. All amino acids having a single amino group and a single carboxyl group and an R group that does not ionize will resemble glycine, because in case of glycine, it has a one amino terminal and one carboxyl terminal. So all those amino acid that has one amino terminal and one carboxyl terminal or the side chain that do not ionize will behave like glycine only. So for all those amino acids, they will have similar pKa value. Not identical, but they will have a similar pKa value. Correct? For glycine, the pKa value is 2.34 and 9.60. 2.34 for COH because at that time COH will lose proton, and this is for NH3 plus because NH3 plus will lose proton around this pH, right? So all those amino acids that have only one amino terminal and only one carboxyl terminal. In their side chains also no amino or carboxyl ends are present. All those will behave like a glycine. So all those amino acid will have a pKa. The lower pKa will be around 1.8 to 2.4 and the higher pKa is around 8.8 to 11.0. But What about the amino acid that has ionizable R group? In that situation, let's take an example of a glutamate. What is the structure of glutamate? So apart from having alpha carboxyl in amino terminal, it is having one extra carboxylant. So now how the graph will work? At very low pH, fully protonated form. As the pH increases, this alpha carboxyl will start losing H. This pK1, that means half of alpha carboxyl has donated proton, half is about to donate. As the pH increases further, at some point this alpha carboxyl terminal will not have H and the donation will start from this carboxyl terminal present in the side chain. Further increase of pH, a point will come when half of this carboxyl terminal of the side chain donated proton, half about to donate proton. So this is pKr. More increase in pH, what will happen? The amino terminal will start losing proton. pK2, half of amino terminal donated proton, half about to donate proton. Clear? So these amino acids can buffer around 3 buffering zones. Around pH 2.19, around pH 4.25, around pH 9.67. Now how we will calculate the PI for these amino acids? Half of pK1 plus pKr, these two. So that is 2.19 plus 4.25 divided by 2. It will form 3.22. How a positively charged amino acid will behave? For example, histidine. So they have one carboxyl terminal, one amino terminal, and in their side chain they'll have positively charged amino acid will have one more amino terminal. So this pK1, that is half of carboxyl terminal donated proton. More increase in pH. pKr, the amino group present in the side chain, half donated proton. pK2, the alpha amino group will start donating proton. So they can also provide buffer around 3 pH. So they also have 3 buffering zones. So how we can calculate PI for these amino acids? It is pKr plus pK2. So for negatively charged amino acid, PI is half of pK1 plus pKr. And in case of positively charged amino acid, PI is half of pKr plus pK2. So this is how we can calculate the PI for any amino acids. Now one question from my end for you all. You have to tell me which amino acid can have buffering potential in intracellular and extracellular fluids of most animals. For this, you should know what is the pH of these fluids. What is our body pH? It is around pH 7.4. So now tell me which amino acid can provide a buffering zone around this pH. For this, you should know the pK1, pKr and pK2 of all the amino acids. If you know that, you can easily tell which amino acid can provide buffering zone near intracellular and extracellular fluid. Okay, do let me know in the comment section if you found the video informative. Please do like, share and subscribe the channel and hit the bell icon for the next video notification. Till then, stay safe, stay happy.